Hi everyone, in this video I'm going to be discussing the steps involved in generating acetyl CoA from pyruvate. So once glucose is broken down, pyruvate molecules are formed. These molecules then go on to produce further energy for the cells. And in order to go from glycolysis to that citric acid cycle, we first have to harvest those pyruvates into acetyl CoAs. So this process is going to involve many small steps, which result in the main entrance to the citric acid cycle. All of these reactions are going to take place in the mitochondrial matrix and again result in the production of acetyl CoA. So this is going to be summed up in this equation shown below. I think this picture does a really good job of showing an overview of what this is going to look like, with us focusing again on pyruvate being transferred to acetyl CoA with the help of a few enzymes and cofactors, and kind of demonstrating how that acetyl CoA is going to be brought to the citric acid cycle so that that can be completed, which is going to be really important for our production of ATP. So these reactions are going to be catalyzed by a complex of enzymes called the pyruvate dehydrogenase complex. And that consists of a total of 3 enzymes, E1, E2, E3, shown there, which I will not do a good job of pronouncing correctly. And then 5 cofactors are going to be involved, thiamine pyrophosphate, abbreviated as TPP, lipoic acid, FAD, CoA, and NAD+, as stoichiometric cofactors. And this conversion of pyruvate to acetyl CoA can be kind of described in a total of 3 steps. The carboxylation, oxidation, and then the final transfer of acetyl CoA. So in step 1, the decarboxylation, a carboxyl group is snipped off of pyruvate and released as a molecule of carbon dioxide, which leaves behind a 2-carbon molecule. And so this takes place with the help of pyruvate dehydrogenase, that first enzyme, and the cofactor TPP is going to form a temporary bond with pyruvate. And so this process is also going to release carbon dioxide as that carboxyl group is removed from pyruvate. In step 2, the oxidation step, the 2-carbon molecule from step 1 is oxidized, and then the electrons lost in the oxidation are picked up by NAD+, which is reduced to form NADH. And the enzyme listed there, E2, is going to help to attach another cofactor molecule, that lipamide, and the TPP from the first step is going to be removed then to form an acetyl group. And so in this third step, it says step 1, that should be step 3, that final transfer of the acetyl CoA, that lipamide is going to be released in a reduced form. So that lipamide cofactor is going to be oxidized with the help of that third enzyme listed there so that it can be reused. And the oxidized 2-carbon molecule, an acetyl group shown there in green, is attached to coenzyme A, which is just an organic molecule derived from vitamin B5, which will form acetyl CoA. And then it's important to note that CoA is sometimes called a carrier molecule, but its job here is just to carry that acetyl group to the citric acid cycle. So again, that an acetyl group is transferred to coenzyme A, which results in acetyl CoA. And so here's that picture, and what we focus on again, highlighted there in that colored circle, is showing what we just talked about, pyruvate being converted to acetyl CoA with those 3 enzymes and their cofactors, and then shown there how CoA brings acetyl CoA to the beginning of the citric acid cycle and drops it off there. This is going to be recycled anytime we have a process of glycolysis. So it could be either anaerobically, where it's always recycled and we just have that pyruvate, or any time that we have glucose in an aerobic environment, glucose will be broken down to form pyruvate, which this whole process can then begin again and is recycled in that way.